Hello and welcome to GeneLab Chats. Today we have the privilege of interviewing Gbolaga Olanrewaju from Ohio University. In his most recent publication, Gbolaga and his team have compared data from spaceflight experiments on Arabidopsis plants in the unique environment of the International Space Station. Through meticulous analysis of protein data, accounting for variables such as light and hardware, they've identified proteins necessary for metabolic functions. This knowledge not only informs strategies for improved potential for space farming, but also contributes to our understanding of the ability to use extraterrestrial landscapes. In this interview, Gbolaga describes how the GeneLab data systems and the AWGs contributed to this publication. Thanks for joining us today, Gbolaga. Could you please highlight the most important finding in your study?
Okay, thank you for granting me this opportunity to discuss the research that we conduct in the light of the distinguished professor, Dr. Sarah White at Ohio University. So in this lab, we probe plant molecular signaling pathways, especially as it relates to gravity signaling. And this usually involves sending plants on space flights to the lower Earth orbit aboard ISS. So several transcriptomic studies have been conducted aboard ISS on plants, but only 4 proteomic experiments have been conducted so far on ISS. And we know protein are the cellular workhorses. They are the effector biomolecules in biological system, not RNA. Therefore, in our study, we compared 4 proteomic studies that have been conducted so far on ISS to identify how each experimental condition affect this proteomic result. So we're able to identify plant proteins that are affected solely due to microgravity in space, those that were affected solely due to spaceflight radiation, and those which are combination of these factors. Likewise, we were able to estimate the extent to which spaceflight hardware and the space experimental setup affect the result of planned spaceflight proteomic experiment. So one of the important takeaways from this study was that despite the experiments having significant variability in the protein expressed across all the 4 data sets, the metabolic pathway expressed in the spaceflight was consistent across all the 4 proteomic experiments. Hence, we can focus wet lab studies on this metabolic pathway for probing plant adaptability for cultivation in space. So that's the major takeaway from that study.
It's fascinating to hear. And I have a second question for you. How did the GeneLab data systems or the AWGs help assist you in your work so you could achieve these goals?
Thank you, Jennifer, for that question. This study idea was actually conceived during the GeneLab AWG meeting at the 2022 ASGSR conference in Houston, Texas. And I remember that I presented the initial result of these at the 2023 AWG Annual Symposium, where I got feedbacks that were substantial to the final paper that we released. Majority of the data analyzed in these meta studies were majorly from GeneLab repository, that is the OSD 38 and the OSD 522. Apart from these, being a member of the GeneLab plant and multi-omics group actually provides me with a platform to discuss research ideas, learn new informatic tools, and get big feedback on happenings in the field. So it really kept me abreast on the latest technology in the field. GeneLab and the AWG have indeed been highly helpful to my research and to the research conducted in the lab of Dr. Sarah White at Ohio University here.
Thank you, Gbolaga. I'm glad to hear about the helpful collaborations you've had. We appreciate the time you spent to share this information with us and with our viewers. And to our audience out there, you can learn more about this work, how to join the analysis working groups, and access the datasets analyzed in this work with the links provided in the description section below. Thank you and have a great day.